Welcome back to vaccine design course. Next, our last step that we are going to do is molecular docking, that is to perform the antigen antibody docking. Now, a molecular modeling technique which is called as the antigen antibody docking is used to predict how proteins, which are antigens, interact with other proteins. Here it is antibody. So how the antigen interacts with antibody to form a stable complex. Okay, so let's go to patchdoc server. Either go to this particular link or search for patchdoc. So search for patchdoc and click on to this one. Okay, patchdoc server. Now when you do that, so you need to click that I'm not a robot, and then you need to go to the server. Here you can see receptor molecule and ligand molecule, where receptor molecule is actually the antibody file. So upload your antibody file. Okay, so choose file and upload from your folder the antibody. So let's take the RCSB antibody as an example, and for the ligand molecule, choose file and take the model 1 alignment modeling. That's our antigen model. Then you give your mail ID. So clustering, let it be default, and the complex type, let's make it as antigen antibody. Okay, give your mail ID and click the submit form button. So this is how patchdoc works. Then we'll wait for the results to come. Meanwhile, we'll go to another server which is known as the firedoc. Okay, go to firedoc server. Click on the first result. Again, same thing. Click I'm not a robot, and then go to the server. Here again, you need to upload your receptor molecule as well as the ligand molecule. For the receptor, it means the antibody. So upload your antibody molecule and as well as your ligand molecule. Okay, so now we have uploaded both the molecules, and then what you need to do is you need to upload the transformations file. For that, you need to get the result of the patchdoc. Anyway, let's go back to the patchdoc result. If it's sent to your mail, open your mail and then go to this particular link, or go to the particular link in which the mail you have been sent. Okay, so when you click onto that link, you will get your results. Okay, you can see the scores, area, ACE, transformations, and the PDB files. You can see so many results, right? So there are so many number of transformations. So what you need to do is you need to download the transformation file. Okay, so click download transformation file. So here you can see that there are a lot of transformations. So here in firedoc server, it says you can upload only 1000 transformations, right? So now, select the number of transformations that you need and then paste it in your notepad. Okay, so I'm gonna select around 900. So let's select. Okay, let's select till 20. Okay, Control C and then paste it in your notepad, right? Okay, 1 second. Yeah, so we've pasted the transformation file. Oh, okay, you can't paste it like this. What you need to do is you need to copy this again. Okay, let's just cut this and save it in a different or a new notepad file. Okay, so I've saved it in a new notepad file and then rename it as transformation file, and it's RCSB as well as the alignment, RCSB and alignment modeling. Okay, that was the receptor and ligand that we selected. So for your exercise, you try out the other servers, Fiery, Swiss model, their automated modeling result, as well as the antibody, other structures that you have obtained, and then see how it goes. Okay, so now I have saved the transformation file of this, and now go to the firedoc server. Now you need to upload the transformations file, right? So choose and then choose the transformation file which we have just now saved. Okay, so what all is been saved? The receptor molecule, ligand molecule, as well as the transformation file is saved now. Next, you need to provide your mail ID here, because all the other things. So this was option 1 that we selected. Output structures, we are giving it as 10. Then give your email address and click the submit button. Let's wait for the results, but before that, let me just explain you about the transformation file. You can, in the FireDoc, there is help button. You can see that a file containing, it's the ligands transformation, right, where we have obtained it from the patchdoc result. So it says X, Y and Z, okay, and which says rotation, Y is rotation, and again X is translation, okay, of all the different forms of it. Okay, that's how it's rotation, translation, and translation. So basically it's the ligands transformation in this following format. Okay, please read it. It's in the same format that we saw. That's what we have copied it. Okay, so here the results usually will be sent to your mail ID. Click onto that link and then you'll see the results. So in the results, the topmost one is the best structure that is predicted, because as you can see, the global energy, that is minimum. So now if you click on download best structures, you'll be able to download all your structures that you have received. Out of it, you will get the best structures. You can also download your solution table that we see here. Then you can also download the refined transformation file. But click the download best structures, because that's what we need. So a zip file will be generated, or zip file will be downloaded in your folder. So you have to copy and paste it, extract the file and open the same thing in UCSF Chimera. If you do not have that particular software, please install it in your PC. It's called UCSF Chimera. It's to visualize the antigen antibody, its interaction and everything. Okay, so now open your FireDoc solution folder. Okay, so let's paste it in our folder named Immunoinformatics, and then you need to extract your files, right? So right click and click your extract here button. So all the files have been extracted. So here in the solution table, you can see there are 10 best structures. So even when you extract it, you can see that there are 10 structures being displayed in your extracted file. Okay, so now open the UCSF Chimera. So this is the UCSF Chimera which is there in my PC. I've opened it. Open file. So here what you need to do is now select option structure, then make it ligand. Okay, so now what you need to do is you need to open the file, right? So click open and open one of your files. Okay, it's there in your Immunoinformatics. So let's take one file. Okay, so this is your solve 1, right? The first model, or first the antigen antibody docked model. Now select structure and then ligand. Okay, so this is here, this is where the ligand is. Now select, then you can go to zone and then you can press OK. It's just all the atoms and everything, but this is your particular type of ligand. Okay, now what you can do is the next one, go to actions, label, then go to residue and go to name plus specifier. Okay, so here when you do that, all the names and everything will be specified here. Now go to tools, go to structure analysis, and then if you want to find H bond, you can find H bond here. Okay, just click OK on it. Okay, see, these are the hydrogen bonds that you can see in this particular model. Now do the same thing by submitting the other files as well. Okay, that is by submitting the Lyra file, by submitting the FIRE2 file. Okay, please do that. Please submit the various receptor models that we have saved, and also various models that you should be saving, because we did not save the automated modeling result. You can take the model, save it, and then upload it and see. Okay, so anyway, this is how you need to visualize your structure. But let me just show you again. Let me close the UCSF Chimera and then open it and show the second model maybe. Or let me just open and take the next file. Okay, let me just take the 10th file. Okay, wait a second. So when you do file close session, the session will be closed, but you can see the files which are here. Okay, so this was again closed session. Let's take the third one. Okay, or let's open it and take the third result. Okay, so now if you want to see the ligand, select structure, then you can see the ligand here. So these are your ligands. Okay, so by this you can see the markers, all the other things. You can see the side chains. So play with this thing. So this is how you need to see. This is your antigen antibody docked model. See how beautifully the ligand is being attached to the antibody molecule. So when you open the structure, what you can do is you can select and take structure. You can actually view the ligand here, which we have already mentioned. Then you can also view your chains, right? Your chain A is being here. Then you can view your chain B. Then this is how you can view your heavy chain, and this would be your light chain. Okay, so this is where the ligand is. Now if you want to see the ligand, you can see the ligand here. So here in this particular structure, this is your ligand and the ligand zones. Okay, can you see? So you can scroll down as well as scroll up to, you can minimize as well as maximize it. So this is where the ligand binds, and these are all your antibody antigen model. Okay, so play with this UCSF Chimera. Help button, go to the user's guide. If you want, you can read about the UCSF Chimera as well. Okay, so this is how you do the molecular docking, and the other parameters if you want to go through, you can always go through by looking at the structural analysis and finding out the H bond and other related things in your model. But please remember that go for the best model to have a nice vaccine. So to get a beautiful vaccine, what you need to do is you need to have an antigenic protein but non-allergic, a stable one. You also need to have a stable antigenic protein structure as well as a stable antibody structure. Now after doing all these things, after alignment, you can view it in certain softwares like the UCSF Chimera, and then beautifully see various bonds, residues, and other amino acid sequences and everything. Okay, so that's about it. Thank you.